Okay, we next have 5 concepts to present to the council. Before we can publish a funding opportunity announcement, we have to present the concept in an open session. We always use the open session of council for concept approval. So we're going to begin with a concept titled Machine Learning and Artificial Intelligence Tools to Advance Genomic Translational Research. And Sandhya Xirasagar, Program Director in the Office of Genomic Data Science, will present the concept.
Great. Well, thank you for this opportunity to present this concept. But before that, I'd like to thank Susan for that wonderful foundation that she has laid in order to present this. And I can see that we can definitely leverage a lot of resources that is being developed at the ODSS. So the concept is ML AI tools to advance genomic translational research, and we are presenting this for concept clearance. I'm presenting this on behalf of Ajay Pillai in the Division of Genomic Science and Rob Rowley in the Division of Genomic Medicine, who have co-led this effort. I'll start with a brief outline. I'll start with the objective and present the background and rationale for this objective and the scope, the budget, acknowledge the help that all my wonderful colleagues have given us, the feedback that they have given us, and then appreciate your feedback. The objective of this is to spur the development of novel machine learning and artificial intelligence tools to explore their potential to advance genomic translational research. Specifically, the aim is to model pleiotropy and variable penetrance through the learning and classification capabilities of MLAI to uncover novel relationships between genotypes and phenotypes. Tools will be developed in a shared, agreed upon ethical, legal, and social implications framework, and I think you see a lot of resonance with the previous talk on this. I'll start with the quick definitions, which I know you're all very familiar with. But we see artificial intelligence as the capability of a computer system to mimic human cognitive functions, such as learning and problem solving. And machine learning is an application of AI whereby mathematical models of data are used to help a computer learn without direct instruction. Now, machine learning has been used for several decades for classification and for pattern recognition. However, with recent advances in computation resources, such as those that Susan mentioned, they're increasingly able to leverage multimodal and large data sets. And in fact, MLAI is being used in biomedical research and in medicine. An example is one where MLAI approaches have been able to discover novel patterns from retinal imaging scans to help detect Alzheimer's disease. Now, the adaptive capability of these tools to use new data as they become available is particularly promising for their potential use to assist in clinical decision making. And an example of that is where MLAI tools trained on detecting high potassium levels from electrocardiogram scan data are being used to non-invasively monitor these potassium levels in, for example, chronic kidney disease patients in the Mayo Clinic in the clinical site setting. What this means is that MLAI tools developed in a research setting are being able to be translated gradually into medicine. We, however, see that we need to invest more in understanding the applicability and feasibility, and importantly, the LC, to understand that of these MLAI approaches in genomic translational research. And that is the opportunity being presented in this concept. So let's take what an example may look like, right? We know that pathogenic variants, even those that are highly studied, due to variant penetrance and pleiotropy, result in phenotypic differences that remain unexplained. Let's take the example of cystic fibrosis, right? We know that more than 2,000 variants, for example, have been discovered in CFTR gene And these result in varying phenotypic differences that are unexplained. And so you can see that in this table here, the pluses, you can see that varying number of pluses are here. For example, with the gene modifiers, with non-CFTR gene modifiers, as well as the environment. In fact, just last month, a paper showed that levels of mercury and molybdenum in the groundwater can affect the susceptibility of cystic fibrosis patients to certain mycobacterial strains. And this, as you can imagine, is a social determinant of health factor. If you look at interesting gene-environment interactions, the ability of secondhand smoke to affect cystic fibrosis is modulated by variants in the transforming growth factor beta-1 gene. We also know that therapeutic responsiveness to drugs that can actually help with symptoms in cystic fibrosis, there is not very well understood. Wouldn't it be great if we could use these MLAI approaches to complement these existing efforts to understand penetrance and pleiotropy, to help us understand the variability so that we may be able to better understand therapeutic responsiveness or sensitivity to different factors. Feedback from recent NHGRI workshops, for example, the genomic medicine, for genomic medicine workshop in 2021, or the more focused machine learning and genomics workshop, have emphasized the need to invest in research to better translate findings from genomics research into genomics medicine. This has also been emphasized in the strategic vision document in 2020. And importantly, the vision also emphasized the need to understand the relationship between genomic and non-genomic contributions, and such as the social and environmental contributions to health. So just a quick recap then to highlight. The objective is to spur ML/AI tool development to uncover novel relationships between genotypes. I keep bringing my cursor back here, between genotypes and phenotypes, and in an agreed upon LC framework. So this is an illustration from the strategic vision document describing the continuum between basic genomics research and clinical care. We see this initiative as a bridge between these two settings. The tools will be developed and validated in a translational research setting by clinical research physicians. And we hope and we think that the successful tools will then, in the long-term vision for these tools is for them to be tested in implementation settings, which means that for this initiative, we do not intend to focus on adoption or implementation science. Coming to the scope, significant investments have been made to generate data sets, and they've just been described: genomics, omics, phenotypic data, wearables, Internet of Things sensor data. Along with these investments, we have also invested in interoperability standards, once again, that was discussed just now. Plus, with advances in computational resources and with data sharing policies, again described before, these have all helped to make helpful in making fair data more accessible. Very importantly, we have generated a lot of reference knowledge. So, for example, ClinGen that Eric in the morning mentioned. So these are all things that we hope, can be used to train ML/AI algorithms such that maybe we can help answer some of these questions with, for example, in the case of CFTR, will we then be able to stratify these patients based on sensitivity to drugs or for susceptibility to infections? Now, for this initiative, we plan to focus on pleiotropy and penetrance specifically, and also to genes with known pathogenic variants for which there is a lot of data that's available. And, for example, the ACMG gene list, that's by no means the only list, but we intend to focus on genes that have a lot of data, diseases that have a lot of data in it. And this is, of course, something that we just brought about before. We plan to make sure that we try to address the LC concerns. And I won't go over the LC concerns because Susan just went over them: bias in data sets resulting in incorrect diagnosis or risk of compromising patient confidentiality. These are all concerns, and we intend to try to address them during the development of these tools. Therefore, we plan to ensure that the tools are developed and validated within a consortium established LC framework. So this is a collaborative project that we believe will be informed from contributions from several researchers. And I don't want to, I know she just talked about trustworthiness and interpretability, explainability, usability. These are all concepts within the LC that we will plan to address. So this is an explorative project. We want to make sure, we want to try to set ourselves up for a more flexible as well as an informed execution. And therefore, we plan to have a phased, milestone-driven approach. And this was also thanks to the Genomic Data Science Working Group, with whom we had a pre-concept discussion, and that working group is actually a working group of this council. We had a discussion with them, and they recommended this biphasic structure. We plan to have a design phase during which the consortium of researchers that we fund, the tool development sites, will select diseases and pathogenic variants depending on the data adequacy of the data to be representative of populations that are affected by the disease, design tool endpoints and outputs and validation plan. Generalizability is an important feature that has been brought up before today about how NHGRI wants to have generalizable tools. And so one of the approaches we are going to use is make sure that the data sets are prepared for cross-validation, that the tools can be cross-validated. And as I mentioned, formulate draft best practices and LC framework. And this will be in phase in years 1 and 2. At the end of the design phase, we will evaluate whether the objectives have been achieved. And if the objectives have been achieved, then we propose to go to the tool development phase. Here, each site will develop tools according to the framework. They will validate according to this plan. Of course, there will need to be refining of these best practices and framework and the plans. They will disseminate resources fairly, and as you can imagine, tools, data sets, the framework, lessons learned. And this will span years 3 to 5. Now you can see that there is a lot of coordination that's involved. Therefore, we propose a coordination center to coordinate the logistics for all of these collaborative activities. And years 3 to 5 of this coordination center will, of course, be contingent on continuation of the development sites. Coming to the mechanisms, we propose 2 RFAs, one to fund the development sites and the other the coordination center, UG3 for the design phase, UH3 for the tool development phase, and a UO1 to fund the coordination center. Relationship to ongoing activities. This is NHGRI's really first initiative to lay the foundation, both methodologically and technically, to develop these MLAI tools in an LC framework. And we definitely, however, plan to benefit by using the data sets that have been generated from NHGRI programs, as well as from the other NIH and global resources. Best practices and LC relevant resources, there are a lot that are generated, and we wish to leverage them. And I mentioned here just a couple of more recent ones that are being put forth. Coming to the budget, Total annual costs of 3 to 4 sites for a total cost of 4.8 million, 1.2 million for the coordination center. Total cost for 5 years of 30 million. Now, this is based on a multidisciplinary team with integrated expertise in MLAI, data wrangling, software development, clinical research, LC, and coordination. Co-funding will be sought from other ICOs for additional sites. Susan B., if you're still there, then probably we'll come to you. Okay. So I'd like to acknowledge the input that we have received from so many of my colleagues, including you can recognize Ken Valley from GenCats. In particular, I'd like to thank Valentina Di Francisco for her help with this, and Renee Sterling for help with the LC initiatives. With that, I would like to get your feedback, and I think Drs. Tim Reddy and Kyle Brothers are the discussants. So thank you.
Great, thank you. I love that this is. I love the ambition here. I love how forward-thinking this is. Some of the things I really like are the opportunity to span across sort of consortia such as Gregor, Emerge, et cetera. I think that's a really exciting opportunity that's really only going to become possible now that we're seeing, for example, more data on Anvil, for example. Right? So I really love that we're spanning those. And I also really, really like the emphasis on validation. I think that's going to be a critical challenge. And the more we can establish best practices and frameworks, especially as the models take in more diverse data and have many, many, many more degrees of freedom, I think that's going to be a really critical point for these consortia for this consortium to address. So I really commend you for taking all that on. I think that's really good. I did maybe just a couple opportunities. One is as you think about spanning across consortia, I noticed a lot of the more basic sciences consortia weren't represented, things like GTEx, DGTEx. That might be some opportunities to kind of integrate with other kind of parts of the NHGRI ecosystem, if you will. It probably wasn't intentional, just sort of, as we're thinking about, getting ideas into researchers' minds, that might be one idea.
Yeah.
And then the second one is, I think it's a really interesting model of this check-in after 2 years. I'm curious how you see that playing out, because I could see I can see applications coming in. I know this gets into implementation, but where you have very different disease areas, but they're each supported by domain expertise, and it might be hard for people to switch to a different disease area. And maybe that's okay, or maybe you're expecting the consortia consolidates on a single disease, or sort of how do you envision that playing out, and maybe like what happens if they don't get a strong consensus?
Yeah, you bring up a good question, and I'm not sure that I can answer the question at this time in that way. But I don't think that we are thinking they should focus on just one disease. But I think really what we are looking for is the expertise and the ability to work together, right? And that mission to make sure that they're going to be able to work to develop cross-validation tools, because that's how we are going to benefit in the end. So we are looking for that eagerness, we are looking, and I'm not sure that I can tell at this time how it's going to go. But I definitely, if you have recommendations, I would really love to hear them.
I mean, obviously, that'll come more into perspective when you see applications, for example. That's an area where I can see there being a lot of challenge with this, and that might be something to just put some additional, planning into as things take shape.
Yep, totally agree.
Okay, Kyle next, and Peter, I see your hand, but just hold on.
Unlikely to be surprising that I'm focused on the LC parts of this. I really think the focus on, incorporating LC into the design of this effort is right on. And you had all the Genomics and Society folks involved in developing this, so obviously their hand is visible there. I think that's really critical for this type of project. I think there's a question I'd love to hear other members of council's opinion on the best way to incorporate LC, right? So my personal view is that worst case scenario is you put out these NOFOs and, you know, everyone goes scrambling to find some LC people and then to put their names on the proposal, and then they're sort of there as technical advisors, but not really any substantive engagement. I think that's a suboptimal outcome in terms of making this a success. I think one way to decrease the chances of that happening might be to incorporate the, you know, requiring specific aims, specifically focused on LC kinds of design, but also evaluation. So this is a whole part of the project, not just sort of a technical advisory kind of role. That's what I see as probably the best way to make this a success, but I'd love to hear what others think, because obviously there are other consortia that have had LC sort of embedded in different ways, and there's, I know, pros and cons that folks have probably seen with those.
I will let others maybe weigh in on this topic before I say anything, or, yeah. But I completely agree with you, so, yeah. Yeah.
1 second, Nancy. Peter, go ahead.
Hi, Sandhya. Yeah, I have two suggestions, and maybe this is there in the actual NOFO text, but it's good to kind of specify what you want. And I notice you used the word penetrance. For me, as somebody who spent part of his life as a medical geneticist, that means Mendelian. But on the other hand, you mentioned data sets such as eMERGE that are typically for common complex disease. So I'm wondering, you know, are you looking for penetrance and pleiotropy in Mendelian disease or common complex disease, or both? And please just put, that should be kind of specified in the text. Another thing that I was asking myself is, why actually use machine learning and AI as opposed to statistics? And would you reject an application that was mainly using statistics? And, you know, given that, especially if this is Mendelian disease, we're likely to have, you know, limited data. So, you know, there's rarely, you know, tons of genomic data and tons of phenotypic data for any disease out there. And so, you know, machine learning might be likely to overfit. I mean, I'm a little surprised that the NOFO is so focused on machine learning and not just let's actually do science and discover things with whatever algorithms possible.
Yeah, good question. So the first question that you mentioned, I don't think that we are specifically honing in on Mendelian, I mean, like at least monogenic sort of disorders. And then the other thing is we are really looking to incorporate diverse multimodal data. And because I think that we stand to benefit from investigating the use of that data, which is why we are considering the MLAI approaches. And because we see that a lot of these capabilities, a lot of these tools are being used, but without taking into consideration the LC and those types of concerns, right? So that is why our focus is using MLAI, I would say. Rob or Ajay, do you guys have anything more to add? But yeah, so Peter, I mean, yeah, I think that's what I would say. Did I answer your question, or did you have more?
I just food for thought, not a criticism, but just suggestions for sort of clarifying the. Yeah.
Right. Okay, but I just wanted to make sure, Kyle, I know you wanted the LC question to be addressed kind of broadly, I mean, with the council members, and I wondered if anyone else had anything to weigh in on that. I'm happy to end that question, I mean, and answer that question towards the end, but if you have anything, please.
Okay, since we're on that topic, I'm going to invite Lisa and then come back to Nancy.
Thank you. So picking up on Kyle's comment, I'll make 2 points. One is to urge that however the NOFO is written, that it is clear that LC concerns are bigger, broader, and I would say more important, but you may omit that one, than being fair with capital letters or trustworthy with capital letters, where that is about the data and where I think LC is about the entire context of data being collected and ultimately used. And then secondly, I think you may want to consider whether you are wanting research to develop an LC framework for these projects or for this data use or analysis. Do you want research? Do you just want a consultation? That doesn't seem to be enough because you really want something new developed. But then I think it's not just a project to develop it, but to assess it, to figure out how it works. Does it work? What are its limitations? What are its advantages? And that way the LC findings will be, I hope, generalizable or informative to others as well, just as the data would be. And then there are different models for this, as Kyle was suggesting. Do you embed one or more LC researchers within the team? Do you do something first? Do they happen alongside? There are different models of that, and probably you want to be agnostic with regard to the model so that different teams could play to their strength. But I think being explicit about whether you indeed do want research on this would be very helpful.
Yeah, very good point. And I think, yes, I think in the end, we want a good framework. We want to achieve our goals, and we want to make sure that they understand that the LC is an important component of this, and whatever is necessary for it to get done, they have to propose the right their right specific aims should address that. Did you want to say something? No. Okay. It is. Yeah, I would agree with you, though. Thank you.
Okay, Nancy.
So, yeah, like Tim, I really admire trying to get out ahead of this, and I think this is absolutely something that needs to be done. That said, you need to be prepared in advance for the fact that you're likely to get grants submitted for this RFA that will be very narrow in terms of phenome, but multi-omics. So not big data sets, not large numbers of people, but deep with multi-omics data, as well as very broad data sets that would have lots of phenome and maybe lots of sequence data and no other multi-omics information, but maybe some social determinants of health. And then you're going to have to figure, and quite possibly, reviewers will find that both kinds of applications sufficiently attractive that you're going to have those apples and oranges coming into this effort. And that can be good because I think there's some reason to be optimistic on some of the deep learning models that we don't need zillions of people. But your specific focus on penetrance and pleiotropy, it means, I don't know how the multi-omics, very small numbers of people address penetrance. Moreover, a lot of the data sets where you'd have opportunities to really validate penetrance are not going to be ones that can be publicly shared and gone back to a lot. So UK Biobank is a big resource, but really healthy people, and a lot of the sequence data available in data sets where you've got a lot of phenome and might be able to really look at penetrance were generated in part by drug companies that may mean that sequence data can't be fully shared. So I think you need to maybe be thinking about this and offering guidance to reviewers. I don't know, but I think this looks like a lot of work for program, cat herding of an unimaginable degree, it looks like to me. So I urge you to be thinking in advance about what you really want with respect to smaller multi-omics, and of course with the multi-omics data that's being generated for the multi-omics project, that's other data that could come into the program. But it's a really I could imagine very different kinds of things coming in, deep and broad, and you need to be thinking in advance about how you would deal with that in the context of a consortium. But I absolutely think, I mean, just the number of people with long histories of machine learning and AI in electronic health records data, the artifacts really come to the fore. And that includes a lot of the crazy biases that we have. There's so many diagnoses that get made only in certain populations. They're completely different names. When a European has effectively sickle cell, it's a hereditary hemoglobinopathy, versus an African American, and it's always sickle cell disease. And all of those kinds of things will come into this broad scale. And so I just can't agree more with what Kyle said. The biases that come in from all of this, you really want to be front and center, and the LC is all about that. So yeah, go Kyle.
Iftikhar. Thank you, Nancy.
I'm still struggling with the numbers. I think Peter brought it up and Nancy, but there's allelic heterogeneity, there's genetic heterogeneity. So I'm familiar with LDLR. There's 2,000 pathogenic variants. And they have different kinds of penetrance because, so I mean, when you might have to study it variant by variant, and then you probably have a handful. If you took all of us, you might have 4 people with the same variant. And then what are you going to do with that data? I mean, you could cluster by disease, but then, as I said, there's 1,000 variants that cause the same disease. So I think you would end up with very few people with a particular variant that you would want to then study. So that's one issue. The other is for deep multimodal data, you might get it, it'd be scattered. Some data sets will have it, some won't, as Nancy alluded to. So other thing that might be really helpful is recall by genotype, which might be possible in all of us. So you could bring those people back and you can really get very deep data, whether it's IoT or STOH or other things that might affect penetrance. But I'm struggling with the numbers here. I would like to comment on that.
Yeah, I can tell you these are good questions, and I think we'll definitely need to think when we write, and especially like Nancy said, the reviewer criteria and so on, and to, yeah, I don't think I have an answer for that, but yes.
You have CFTR. I don't know this. I am not a pulmonologist, but it's like 2,000 variants in CFTR gene.
Just a small comment. That's the reason why we're doing the 2 phases, right? Because that's we know that we're going to have all of these questions you're all raising. Those are all excellent questions, but we really need to try it out and see how good it is or how bad it is in our data sets. I mean, maybe,
Yeah.
Think of variant annotation as the first target instead of penetrance. I mean, there's a huge need for variant annotation. I know there's a huge investment in it, but I think machine learning could really offer a whole lot. I don't know whether that's something to consider before penetrance and pleiotropy, is to actually annotate the variants that are coming out in droves with all the sequencing.
Do you have any?
I mean, just a reminder, every amino acid in insulin has been mutated to every other possible amino acid. It's a small protein, and lots of those are now products that are used. But when those were examined in cell-based assays, they nearly all have function. But we've seen lots of them in people who are fine. I mean, if you bring them in and do exhaustive physiological studies, you can see some of those things. But phenotype is not necessarily, cellular-based phenotypes are not necessarily telling you about penetrance. And so it's a people versus assay sort of thing. There are lots of data sets now where people are, mutating things and doing RNA, looking at downstream RNA-seq, and that's data that could be used for these kinds of things. But it's a really complicated question. I mean, just the concept of penetrance, like Peter said, it's a really fraught thing.
Yeah. Did you want to say something? No. Yeah.
All right, thank you for that discussion. I'm going to call for a vote on the concept. Can I get a motion to approve the concept? All in favor? Any opposed? Peter, you're opposed or you're just late?
Sorry, I'm in favor. I was late.
Okay, very good. All right. Thank you, Sandhya.
Okay, great. Thank you.